Good morning, everybody. Welcome to this webinar. We will discuss about biological quality of soil and plant efficiency. This is the agenda for today. First, a brief introduction from me, then Lin Bautze from FiBL and the project Climate Fab Demo, and then we will leave the floor to Professor Cristina Menta and Mariolina Gullì for their speech. Just a brief presentation of the two sister projects where we are involved. Climate Farm Demo involve a quantity of farms all over Europe, and they are pilot farms to show the best practices about reducing climate impact for agriculture. And the other is Climate Advisor. Everybody can participate in it. It's free, and it's for the formation of advisors and consultants in good practices against the climate change. The two projects are divided in thematic areas, and this webinar is organized for soil, earth, and biodiversity thematic area, by the thematic area of soil, earth. I will discuss about discovering a multidisciplinary approach to soil quality, and as you see in the bottom, you can, for Italian agronomists, there is a message because we can gain credits information participating in this webinar. I like to remind that it's just a matter of age. Soil is the heart of the life on Earth. Times ago, we think that soil was just physical, due to physical disintegration and chemical alteration of rocks, just a little bit influenced by atmospheric conditions and living organisms. But now we must change our perspective. Soil is the most valuable resource of humanities, and it's through soil that plant can work. And if plant can work, we can live, because plants produce the entire food supply, directly or indirectly. Soil is a complex living organism, so it's not possible to think that we can understand soil just using the classical chemical physical analysis. We must discover physical properties, like qualities of its structure, porosity, root depth, through, for example, a survey that is called visual soil assessment, and we'll discuss it in the future. And of course, to discover biodiversity. Sorry, just a little bit. Okay. And today we will discuss of this. Soil biodiversity contains the metabolic toolbox that support pedogenesis. So without biodiversity, we have no soil formation. It doesn't exist, the soil, without biodiversity. It cannot exist. And as well, it's critical, the biodiversity, for sustainability of the ecosystem functions. For example, nitrogen cycling, organic matter decomposition, and plant production. So without biodiversity, we don't have ecosystem services, and the soil doesn't work properly. This is just an example of several surveys that we manage all over Italy to understand the function of soils and the importance of biodiversity in soil, using the QBS-RR index that we will discuss during this webinar today. We perform more than 1,000 samples. The data are from Sata Studio Agronomico and Fondazione Edmund Bach, and the statistical analysis was performed by Agri-Food Research Hub of University of Brescia. And this is just one of the results. For example, the relationship between QBSR, so the biodiversity of soil, and total copper. And there is no linear regression, so no effect between these two data indicators. Of course, this doesn't mean that there is no effect either, but we can use this information to understand which is the practical utility of these surveys. We know that we must be aware of what is occurring in the rhizosphere. And if we are aware, we can define the best agricultural practices. So thinking to new strategies, applying the strategies and checking the results during the time or in several different environment and agricultural systems. So if we reduce the impacts of our activities through good strategies, we can increase the resilience of the system. Resilience because, as you know, as we told before, it's a complex system, so we can speak about resilience because it's a lot of different things that happen in the rhizosphere. And if we use the good strategies and we know what happened when we apply the strategies, we can increase soil health, biodiversity, and of course quality and quantity of production, because earthier plants that live in earthy soil can produce much and better, and plants longevity. And today we will discuss also of another matter. As soil is complex, plants of course are complex, and so plant behavior depends from interactions among different things among themselves, environment and pathogens. And to have a good decision-making process, we must know what happened and try to understand what happened, but we cannot presume to switch on off a problem as if it is a switch. So using biostimulants, it can be a good result, it can be a good strategy, but we have to understand if they are efficient and how they work. Maybe sometimes it's very difficult to understand how they work, but we can try to understand if they are useful. So this is a tentative description of biostimulants, formulated product of biological origin that improve plant productivity as a consequence of novel and emergent properties of the complex. And so the second part of this webinar will be dedicated to this matter, to this issue. At the end, we as consultant and as advisors, we have to change our way to act. We must be aware that knowledge arise awareness, and soil health and plant behavior is the focus where we work. We must work, thinking again how to approach technical production and strategies, believe in the possibility to be sustainable and share knowledge with farmers, because it's the only way to increase the sustainability of our action through the action of the farmers. So we are ready to leave the floor to Lin.
Thank you very much. So I will share my screen and Now you should see it.
Yeah. Okay.
Super. Yeah, so when Marco invited me to join with the Climate Farm Demo project, where we also have the theme lead of soil health and biodiversity, he wanted me to put an emphasis on the connection between managing soil health and also then the productivity that follows out of it. And then coming from the Research Institute of Organic Agriculture, I really tried to reconsider what the origins of organic agriculture are. And actually, one of the main 4 pillars is associated to soil and soil health. So I would say it's something which is really at the core principles of organic agriculture, to really look at this understanding of how soil health impacts the whole the whole farming system and the whole productivity. And you will often find at organic farms that a lot of the farmers are really taking care of the soil health and really want to understand soil health. And Marco just put it beautiful, like that the soil is a living organism. So that's something which is, I would say, really embedded in the organic principles. And if we look in more detail, like how managing soil interacts or what kind of implication it has in the different areas, we can see that on the one hand, we have the implications for the organic substances, which would be the humus content and the soil organic carbon, which is mainly related to the water holding capacities of the soil, the nutrient availability. But then we really also have this huge part of the soil microorganisms and the living beings in the soil, which is also very much related to soil health. And then, of course, we also have more the structural parts, really understanding how water retention is influenced, or erosion regulation, or soil aeration. But all of them are interacting with one another. So it's really important to find practices that positively influence all of these aspects all together, to really not miss one of the factors, but to understand that the soil is a complex system that needs to be managed also accordingly. And at the Department of Soil Science, where I'm affiliated to at the Research Institute of Organic Agriculture, we have a co-managed long-term trial at DOC. It's called the DOC, so it's existing since 1978, so quite some time, and it's actually a really nice system comparison trial. So it really compares the effects of crop rotation, fertilizer management, and different interactions between plants and the soil and so on, between the 3 main systems of biodynamic agriculture, organic agriculture, and the conventional agriculture system. And it's, as I said, it has been running since 1978, so quite some time. And collecting data during this time really showed us in the research that the organic practices really have beneficial effects on the physical and the chemical, but also on the microbial and the faunal effects. So, for example, we see that the aggregate stability of the biodynamic and organic farming systems really outperform the conventional one. And also for the organic carbon, then we recognize that the organic carbon sequestration is more was higher in the biodynamic and organic parts. And also, if we look at the biodiversity, we see that often the biodynamic and organic systems really support biodiverse, rich soil structures. So when we come back to the context that we meet here, so this is the Climate Smart Advisory Projects and also the Climate Farm Demo project, we realize that the soil organic carbon is also an essential part for really considering how the soil health of a certain farm is looking. And I won't go into detail, you can read that up. So we have provided a nice fact sheet on the relationship of soil and climate. But I would like to emphasize that we see in the DOC trial that the soil organic carbon sequestration really takes time. So it's not something that is like built up immediately. but we see it takes about 20 years to really constantly increase and then kind of stabilize around the system, where we also see that the again, the biodynamic agriculture outperforms the others. And if we look at the effects of this, we can see that the soil organic carbon content then really advances all of the other aspects. So like better aggregate stability of the soil and reduced soil erosion, increased soil water holding capacities. We also see this higher microbial activities, which then also lead to better nitrogen availability and increased microorganisms, so overall better soil biodiversity. And you can imagine, so we see that this is part of a climate change mitigation aspect when we look at soil organic carbon, although there I'm a little bit skeptical on really putting soil organic carbon as a real solution for climate change. But I would say all of these factors, they also highly impact the climate resilience, so the adaptation part of a farm. And that's becoming even more relevant now for the farmers to have a system where their soil health and their productivity in the long run is still kind of ensured when we have considered the unavoidable climate change effects. We have wrapped up these results of the dog trial also in a publication. So if you are interested in further details, it's also nice to read that publication. We have also put emphasis on the different climate effects. So really like the total emissions are lower in the biodynamic and organic systems compared to the conventional ones. So we see if we are doing measures that really protect the soil, that increase the soil organic carbon and the microbiome and so on, we see that on the one hand, we have a positive climate mitigation effect, but we also see really that climate adaptation is promoted more for the farmers. And now I would like to go more into detail how to do this, or like why it is good to really consider this aspects of organic substance and soil microorganisms and soil structure. And I would like to go to the different parts. So for example, when we look at what kind of management systems really promote to increase the organic substance, so the humus, we will find that, of course, the organic substance is related to the climate you are. So it depends on where your farm is located or what kind of soil type you have. But it's also very much related to your residue management or your manure and fertilizer application. So it is actually something that farmers can promote on their soils and strengthen with different practices. And the same applies when we look more into this microorganism and soil biodiversity aspects. So for example, the AMFs, which is one of the oldest fungi you find in the terrestrial systems, they are mainly responsible for the uptake of nutrients and water, so they have quite a relation to productivity. They are also relevant for soil structures, like preventing erosion or supporting erosion control and increased water retention capacities. But they are also important for the defense against pathogens. And you see that they can be either supported, for example, by the application of AMF products, but some of them are also challenging. So it's not so easy to apply them because they need to be local specific and so on. But they can also be supported, we see in our trials, via the indirect management. So of course, like if you avoid chemical substances, if you go for higher plant diversity and crop rotation, or if you reduce your tillage management or even practice an organic agriculture, you can positively influence that your AMFs are more at your place. And the same applies for the soil structure. So you can promote your soil structure or enhance your soil structure by applying organic fertilizers, again, higher plant diversity and green fertilizer, constant soil covers, reduced tillage management. So a lot of these practices that are practices in organic agriculture. And I'm not saying this to convince the whole world that they should shift to organic agriculture. I know in some cases this is not realistic, but still I would like to urge that if we are looking at the soil, it's important to really consider that the soil is essential for a lot of functions, and organic agriculture offers a system that has a lot of co-benefits. So for the soil structure, for the soil microbiome, for the soil organic carbon. And by this, actually, it can combine a kind of climate change mitigation effect, but also prepare farmers really for the unavoidable climate change effects. And as Marco said, so the biodiversity is really a key for protecting the future of our soil health. And from my experience, it's often also a good start to do climate advisory work on the soil topics. So a lot of the farmers are already engaging with their soil management, and they find it quite important. So it's something where we can, as a climate advisory network, really come into a discussion with them. And so I would really say that if we want to do this and come more into the discussion of a healthy soil and a good productivity, then we really need to strengthen also this advisory expertise on the different soil health and productivity patterns, because of course the implementation of what kind of practices work and where a farm is located and what is technically, financially and also socially feasible for the farmer is very different. So that needs a lot of training, and that's why I'm really thankful that the Climate Smart Advisory Project is really collaborating also with the Climate Farm Demo Project and having these discussions like today. So if there are questions later, these are my contacts. I'm mainly working for Climate Farm Demo, Clean Farms and Organic Climate Net. But before saying real thank you, I would like to hand over then again to the next speaker so that we can have our discussion later continued.
Thank you very much, Lin. And now we are ready to leave the floor to Professor Cristina Menta. Of course, I remember that the webinar is registered, so you can share the link in the future to everybody else.
Okay, thank you, Marco, for your presentation. Can you see my slide, my presentation? Yes. Okay. Good morning, everyone, and thank you to the organizer for inviting me to discuss about this important topic, the role of soil fauna in evaluating soil health. And we know that in most cases, people don't even realize that the soil hosts a rich community of organisms that live, reproduce, and perform many essential functions with it. In effect, soil organisms play a vital role in delivering key ecological functions, including nutrient cycling, organic matter decomposition, or the maintenance of structure, the regulation of pests, and so on. And these functions form a foundation for the provision of ecosystem services. However, before discussing soil fauna and their roles, it's important to first recall the essential services that the soil provides and that soil organisms sustain these services provided by soils. And for example, supporting, provision, regulating, and cultural. But soil is the environment where biodiversity, soil biodiversity, found the best site, the best condition to live and reproduce. And we have to consider soil biodiversity not only as the number of species in the soil, but also relationship between these organisms and the functions that these organisms have in the soil. And we know that each member of the soil team performs a specific function. We can see in this slide the number of organisms that we can find under a footprint, and only the cooperation of all team members is able to guarantee the maintenance of soil fertility and the diverse range of ecological services which are provided. So in this slide you can see the difference between fauna in relation to the dimension. In the soil we can find organisms really small, such as microfauna, in particular protozoan or some nematodes, some rotifers, tardigrada, and some mites, acari. But the major part of arthropods are represented by mesofauna, organisms with the range of the body between 0.2 millimeters to 10 millimeters. And then you can see in this case that we have the major part of arthropods, such as sprinters, colembula, or myriapods, or diplura, protura, or many insects. And another part of fauna is comprised the macrofauna and the megafauna in some cases, where you can find some animals such as kilopods, the centipedes, or snails, or some beetles, or earthworms. And all these organisms perform some functions in the soil. And in particular, for example, the organic matter fragmentation and partially decomposition, or they are really important for the soil aggregation and for the creation of the porosity in the soil, for the regulation of water, for example, or they take part in many processes such as nutrient cycling or carbon and nutrient regulation. And we know that abundance, species diversity, biomass of all these animals are really influenced by a broad range of human practices. Human activities normally cause degradation and pollution of soil, and we lost biodiversity, with the reduction of soil biota in terms of abundance and biodiversity, or very often the simplification of soil living communities and the loss of ecosystem services obviously provided by soil biota. And agriculture can be the difference, because for agricultural ecosystems, for example, tillage or treatment of pastures and crop residues, of the use or, for example, of pesticides, fertilizers, manure, soils, and so on, or the use of cover crops can be the difference. And I show some case studies after in my presentation, and you can see the differences between conventional and the conservation agriculture. So we can do the difference in agricultural ecosystems, considering some good practices, for example, organic fertilization, the use of manure or compost, or really important, the crop rotation in contrast of the monoculture, or the maintenance of soil cover with the use of cover crops, no bare soil, or for example, no tillage or the minimal tillage in contrast of normal deep flowing normal tillage. With these good practices, we can improve biodiversity, or using chemical fertilization, monoculture, and so on, or bare soil in particular, we depress biodiversity in agricultural ecosystems. So in this slide, you can see an example and an experimentation that we conducted some years ago with the Veneto Agricoltura in the north of Italy, where we compare the effects of conservation agriculture, no tillage, and the use of cover crops with the conventional agriculture, tillage in particular, and bare soil, on earthworms. And in the first graph, you can see an important difference between conservation agriculture in blue and conventional agriculture in red in three different farms, Diana, Sasserami, and Valle Vecchia. But in my opinion, in my experience, experience is really important. Consider not only the total number of individuals, but also the presence of juveniles. You can see in the second graph that the numbers of juveniles is higher in conservation agriculture compared to conventional agriculture. And these results are really important because it shows that we have the community really able to survive, to continue in terms of new individuals. So, in another experimentation that I show in these two graphs, you can see the effects of conventional agriculture. Conservation agriculture is the same tillage and no-till and cover crops on mice, acari in the first graph, and sprinters, collembola in the second graph. And in this case, you can see that for some crops, for example, for corn or for wheat, we have differences for mice, and these differences are not so evident, in particular for corn, for sprinters, for collembola. Really interesting is the fact of the presence of bare soil compared to crop residues or cover crop, that in both cases, in particular for cover crop, increase the presence of mites and sprinters. So, but we have to consider another important topic that helped me to introduce the QBS-R index, the adaptation to soil of soil fauna, because we can observe a particular type of morphology in organisms, in particular in this case arthropods that live into the soil. In particular, they show, for example, the reduction or loss of pigmentation and the reduction or loss of eyes. And this is because we don't have light in the soil, and for example, really pigmentation is not important, or obviously the ice is not important in the soil. And we can observe in some cases a streamlined body form and the reduction of hairs, antennae or legs, some characteristics that are really, really important outside soil, but in the soil they lost the functions, or in some cases it's important to move inside the really small pore present in the soils, and the evolution brings these animals to adapt in this particular environment and in these particular conditions. We can observe the reduction or loss of flying, jumping, or running adaptation for the same reason, and the decrease of water retention capacity. And the adaptation to soil of these animals makes unable to survive outside heat. And as a result, they are extremely sensitive to changes in the soil's physical and chemical properties caused by natural processes or very often human activities. So, and this is the idea on which is based the QBS-R index, the Soil Biological Quality Index, based on microarthropods community. And this index was developed more than 25 years ago at Parma University from Professor Parisi. And the idea, the concept of this index is that a higher diversity of microarthropod groups, well adapted to soil, is found in soils of good quality and good health. You can see different sprinters in the bottom part of the slide. You can see an epigeous surface dwelling form or an oedaphic form. And you can see the differences between some traits, for example, antennae or legs or the colors of the body. And the epigeous surface dwelling form needs this color and this structure to survive outside the soil. And the oedaphic form presents this type of structure, body form, in relation to the adaptation to the soil. So the QBSR index considers 4 different phases for the application. The phase 1 is the soil collection. The phase 2 is the arthropod extraction. The third phase is the determination of biological forms, and the last one is the QBSR index computation. So the first phase is really, really important, soil collection, because if we make a mistake in this phase, we can invalidate the sample and all the study. It's important to consider the period of soil collection, because animals in summer or when the temperature is too hot or to, for example, the condition of humidity isn't not so good. The animals go in extivation or hibernation, and for this reason, it's impossible to extract these animals because we use a dynamic extractor. So in Italy, for example, the best period are spring and autumn, not in summer and not in winter. So in the index, consider 3 different replicates for area with the dimension of 10 per 10 per 10 centimeters and more or less 5-10 meters away. It's really important to take at least 3 replicates because animals, arthropods in general, have aggregated distribution, and for this reason it's important to consider several points for the collected replicates. The field activities are simple. In the first moment, you have to cut the grass from the soil surface. After, you can use a spade or a soil sampler to collect soil and put the soil in the bag with more or less 30% of air volume inside, and after label the plastic bed, obviously with some information, sampling area, data, and so on. In the lab, it's really important to bring the sample in the lab within 24 is better or 48 hours after collection. You have to maintain the soil sample safe, not under the sun or where there is a not good temperature. After the soil sample to put in the equipment for the extraction of microarthropods, such as the Berlese tool grand funnel or equivalent arthropod extraction tools. There is a lamp for create a dry condition in the first layer of the soil. The Berlese comprises a sieve of 2 mm mesh and 20 centimeters in diameter. Funnel, plastic or glass, is the same, and you can see that under the funnel we have a container, a collection container with 2 parts of alcohol and 1 part of glycerol. And the procedure comprises place the soil sample in the sieve of the funnel, and the extraction time is 7 to 10 days in relation of the presence of water, the humidity of the soil sample. The phase 3 is the termination of biological forms. And this is a really important and, in my opinion, delicate phase because microarthropods are separated following biological form approach, overcoming the difficulty of identifying the species of Fauna. You can see in this table that let's consider the orders of arthropods. We, in particular for myriapods, Chilopoda, Diplopoda, Pauropoda, Symphyla, in the past, the classification of these animals was order, and now they are considered class, but in the QBS, at the moment, we maintain this level of taxonomy. So for each group, ecomorphological indices, EMICE indices, was applied, and you can see that we have a range for this EMI score. The EMI score can range between 1.5, 1.10, or 120, or can be 1 or 5 or 20 or 10 or 20. And this score is in relation to the level of adaptation to the soil of these groups. And in particular, IMAI 1 is no adaptation, IMAI 5 or 10 is intermediate adaptation, and when we discover animals with the maximum adaptation, we give them IMAI 20. IMAI 20 is a total adaptation of the animals. And the phase 4 is the QBSR computation. That is the sum of the highest IMAI values recorded for each taxonomic group present in the sample. In this table, you can see an example of the QBSR application, and we have the 3 replicates, replicate 1, 2, and 3, and the score of animals that we found in these 3 replicates. And generally, at the end, you can observe the QBSR value. In this, with this data, you can notice some differences between the QBSR value. We have 182, 132, 180, and these differences are normal because, as I said before, the animals are aggregated distribution in the soil in relation to the soil organic matter content, in relation of the soil humidity, and so on. And for this reason, we can have differences between the 3 replicates. Generally, in the published papers, we have we consider, not only us, but the researchers consider the mean of these replicates, the average, but you can decide to calculate the QBSR maximum that you can see in the 4th column, where in this case the QBSR maximum isn't the sum or the median of the QBS or the IMAI score, but is the presence, only the presence, the maximum value of the IMAI score that we found in each replicate, but repeated only one time. For example, for Pseudoscorpiona, we have the Pseudoscorpionida only in the replicate 1, we consider with IMAI 20 as IMAI score, we consider 20. Or, for example, we have Hymenoptera in the replicate 3, we consider 5 in the QBSR maximum. Obviously, generally, the QBSR maximum is higher than the 3 replicates, but in my opinion, it's a good, it's interesting because the QBSR maximum gives us information of the potentiality of the area, the maximum expression of diversity and quality, in this case for the QBSR index, the maximum quality of the soil that we found in in an area. So, but we can use IMAI score also for other considerations. For example, in this case, you can see the percentage of taxa that express the IMAI maximum, 20 for some crops. And in particular for wheat, we can consider only the sprinters, the Collembola group, where we can evaluate the differences between, in this case, for example, conventional and conservation. And you can see in this graph that in this case we have a higher number of sprinters with IMAI 20 and 10 in conservation compared to conventional agriculture. So, and the QBS is having a good importance and a good use in the last years. Some years ago, we found a working group inside the Italian Society of Soil Science, and the Food and Agriculture Organization of the United Nations expressed interest in this index, and we are preparing a protocol, standardized protocol for the FiBL organization. But we have to consider not only the weakness aspects, but also the strength aspects. In particular, for the weakness, we have that taxonomic level at the order is really useful rather than at the species level. Is more simple. It's better to consider order and not the species, because for all arthropods it's impossible to arrive to the species level. Or we need more experts in this field. QBSR is low cost and generally fast soil sampling, and the equipment to be used it easy to assemblage, in particular the Turgan funnel. Relatively simple to apply, and really important, the operator can be trained within a few days, 2-3 weeks, with good training. But we have strength because for arthropods we have spatial variability or seasonal effect. It's really important to collect soil in the correct season or in the correct temperature and in general environmental condition, or the vegetation development effect. For example, for wheat, we discover differences between the first phase of the crop and the last phase of the crop. And this is in relation to the presence of vegetation, the presence of a rhizosphere, or the content of organic matter, and so on. And the extraction after 7-10 days, and it is relatively long. So, where the QBSR index has been applied, we applied the QBSR index, not only we, but in general. The QBSR index has been applied in different type of woods, beech forest, oak, conifers, different managements, or after catastrophic events, storm or burn woods, in permanent grassland, orchards or vineyard, the different agricultural crops, with tomatoes, corn, alfalfa, and so on. Or comparison between organic and conservation agriculture, urban parks, or for the evaluation of effects of sludge, covered dams, and the reclaimed area, or many studies are conducted in contaminated soils, to metals, hydrocarbons, and so on. And in this case, I show two experimentations. Where you can see the QBSR value. In the first experimentation, we compare conventional agriculture, tillage, and conservation agriculture, not tillage and cover crops. And you can see the differences between the two managements for different crops or comparison. In particular, you can see that in the major part of these results, we obtain the QBSR value higher in conservation agricultures that in conventional agricultures, but in some cases, for example, in the last two columns, you can see that wheat didn't show this type of results. We have a comparison in these in this crop in the two conditions. In the second graph, you can see experimentation of the application of QBSR with the conservation and conventional agriculture, and the QBS in this case answered good or partially good in different years of the experimentation. So the QBSR has been applied in Emilia-Romagna region, really is applying now here, but in this case we consider the results from 2015-2020 in different cities of the Emilia-Romagna region. And in the graph where there is the box plot, you can see the differences between, for example, arable land, agriculture environment with, for example, rotation meadow or permanent meadow, or vineyard and orchard. And you can see that arable land, where we have corn, wheat, and so on, we have the lowest value compared to the other condition. And now I want to explain an experimentation that is in progress. In particular, we have a project where we are evaluating the effect of the presence of solar panels in agri-photovoltaic system, not the photovoltaic system, but agri-photovoltaic systems that combine the production of energy, clean energy, with the production of food of crops. And in this project, we try to link the aspect about biodiversity with the water dynamics with the economic approach. So in this case, we consider two different crops, wheat and tomato. For wheat, we have 4 treatments in 2 sampling phases, December and May 2024. Crops with 13% of ground coverage ratio, crops with 41% of ground coverage ratio, full light crops without a panel, and control. For the tomato, we have only 3 treatments, crops with 13% ground coverage ratio, full light crops, and the control. And I show really briefly the results that we obtained. So consider in this case in the graph only the different capital letters, and you can see considering the total abundance of arthropods, you can see differences between the control in green and between the treatments with panels and without panels, FS, full sun. Differences between the treatments aren't evident. In this case, we don't have differences between the presence of panels and the full sun, but we have differences with the control. And the same results we obtain for the numbers of orders of arthropods, and in particular for wheat, and for the QBS R value, in particular for wheat. So in this case, we can see that the effect isn't the presence of panels, but more the presence of crop, the effects of tillage or the bare soil in some period, and so on. The presence of wheat in this case, or in tomato, but we don't have evident results in this case. So some years ago, we decided to, in particular in 2018, we decided to collect all the QBS-R papers published and try to the metadata analysis considering different categories, 8 group pages: agricultural lands, soil interested in human degradation, permanent grassland, pasture and meadow, soils in natural degraded conditions, orchard, plant remediation, restore pit mine, urban parks, and woods and forests. And in this graph, you can see that in particular agricultural lands, soil interested in human degradation or in natural degradation show really low value of the QBS. For the urban parks, in this case in particular, the data were not enough, in my opinion, and we need more data to understand if the urban parks show this type of really low value of the QBS, because this experimentation was, in my opinion, not so good about in particular for the application of the index, the methodology of the index. So, and now we are trying to do more or less the same, they use the same approach. We collected all the, I'm not sure all the, but the major part or all the papers that we found in the literature about the QBS-R application, and from 2002 and 2025. And the data set comprises more than 100 observations covering a wide range of land uses and the geographic regions. And but this study, this manuscript is in progress. So we hope that a publication will come out in the next few months to give the possibility to consider eventually the new line for the consideration of good QBS, R value, and consequently good quality of soil, and bad condition and bad quality of soil. So I finished my presentation. Thank you very much for your attention. If you need more information about the application of this index, you can write me at this email, cristina.menta@unipr.it, and you can join us in the QBS working group if you are interested. And I'm happy to know your experimentation, your results. If you have published paper, please send me, because for us it's really important to follow the development and the application of this index. Thank you very much for your attention.
Thank you, Professor Cristina. I can confirm that at field level, the QBSR is enough easy to use and very, very useful to understand deeply how our activities impact on biodiversity of soil. So I encourage everybody to use QBSR index because it's very, very useful. Now we can leave the floor to Professor Mariolina Gullì for a speech about biostimulants.
Hi, everybody. I'm sharing my screen. Please confirm that you can see it.
Yes.
Can you see the presentation?
Yes.
Okay. Thank you very much about this possibility to talk about the use of biostimulant, microbial biostimulant in agriculture. It's a pleasure for me to be here today. A short overview of what I've been talking about, few words about sustainable agriculture in EU, the use of plant biostimulants and their regulation, what are microbial biostimulants, and I will show you some example from field trial that we carried on in the last few years. Sustainable agriculture is a complex system with the three main pillars about economic sustainability, environmental sustainability, and the social sustainability, which has the main purposes to improve competitiveness of the agricultural sector in Europe, to protect the environment through the preservation of biodiversity, managing soil health and water quality, and of course there is an important part about social sustainability, which includes the promotion of employment in agriculture, but at the same time ensure food security for all the citizens. So a focus on environmental sustainability because the agricultural system it has an impact on the environment due to the intensification of farming practices. We all know that the use of input can have, let's say, negative pressure on the environment. First of all, for the excessive use of chemical fertilizers and pesticides. But at the same time, the fertilizers in the last century ensured the food security for all the people, and nowadays we are facing many drawbacks about it, due to the political situation. In some cases in EU, we are in difficulty for the imports of this kind of products. And at the same time, there is drawbacks about food safety because fertilizers and pesticides may have some contaminants or toxic compound that enter the food chain. So it's quite a complex picture that we may solve. And the EU Green Deal targets by 2030 2030 aim to face this problem. So we have this target to reduce the chemical pesticide and the more hazardous pesticide by 50%, and to achieve the 25% of agricultural land under organic farming practices. All of these targets will have the purpose also to reduce the synthetic fertilizers by at least 20%, preserving soil fertility. So soil is always in the focus of these targets. For this reason, I want to point about plant biostimulants, because they can be considered, let's say, a smart solution. So plant biostimulants are defined in the European regulation on fertilizers, which is in charge. Actually, its efficacy is started by 2022, and they are defined as organic or inorganic products which contain bioactive substances and may contain also microorganisms, which applied to the plants or to the soil can stimulate growth and productivity through some specific activity, which is the improvement of the absorption and assimilation of nutrients, increasing the availability of some nutrients which are stocked in the soil, and they can improve tolerance to both biotic and abiotic stress, and in the end they can improve also the quality of the products. They are classified in different in 5 classes on the basis of their origin, composition, and mechanism of actions. And there are 5 major categories which are included in the directive, which are seaweed and plant extracts, humic substances, hydrolyzed protein of different origin, from animal or plant sources. They can be inorganic beneficial elements, but today I will talk to you about the beneficial microorganisms, which are bacteria, yeast, and filamentous fungi, and their function is exerted through the symbiotic association with plants. In this sense, we can say that microbial biostimulants are considered a sort of biotechnological application, which is using knowledge from what happens in nature. Actually, plants living in an open environment, in natural environment, start interaction with several microorganisms that can be neutral, beneficial, or pathogenic. So there is a complex interaction between plants and its environment in terms of biotic association. And the knowledge of this complex network occurring in natural environment gave the possibility to develop new products, which are those called microbial biostimulant. Their action is based on direct and indirect mechanisms, since directly some bacteria, which are called generally plant growth promoting rhizobacteria, can help plants and can improve soil characteristics, because they can fix nitrogen, they can produce phytohormones, they can solubilize and mobilize the nutrients in the soil, which on the whole can improve plant nutrition and at the end enhance the yield. In the indirect mechanisms are based on some metabolic properties of these bacteria, which can produce useful substances such as siderophore for iron fixation. They can induce resistance against pathogenic form, so they can act as a biocontrol agent, and they cooperate, improving also plant metabolisms, so they can give some input for improving stress resistance in general, which determines against an increase in yield. So these mechanisms can act all together. So it's a very complex network, and it has been studied a lot since many years, and so we know a lot about this interaction, but a lot is still unknown. Nevertheless, there are some products already available on the market which are based on some peculiar activity. So we have nitrogen-fixing bacteria. The most famous one, of course, is the rhizobium, which interacts with the legume's root to fix atmospheric nitrogen, but also we are living bacteria, azotobacter and cyanobacteria, that can be very useful in this sense. We have fungi, arbuscular mycorrhizal fungi. As we heard before, they can create this symbiotic network, and there are plants which actually need this symbiosis to uptake phosphorus, for example, and the interaction of mycorrhiza with the root plant can be negatively affected by strong and conventional agricultural practices, and they depend also on the use of fertilizers and fungicide in the soil. So the agricultural practices can negatively affect this aspect. Then we have trichoderma species, which are as a good example of biofungicide. Is a sort of biocontrol agent against soil pathogens. So its presence close to the rhizosphere, the roots of the plant, can protect against many pathogens. We have also bacillus species, which are widely utilized in many sectors, including agriculture, and they are useful in hydroponics as well. And they can act as a biocontrol agent and also can improve tolerance, for example, to salt stress. So there are many information about them. Most of this information have been obtained in vitro, in experimental setup, which is in the laboratory. So we know that they can solubilize phosphate, and they are very useful for the uptake of phosphorus in those soil which are very rich of phosphorus, but the phosphorus is not available for the plants. They can, through the release of acid polysaccharide and enzyme, they can make the potassium available for plants, and they can improve iron uptake as well. So they have a specific action. Each strain has a specific action which has been characterized in the lab. But the focus of today is how can we, let's say, use them in the real situation. So there is a step of production. There are many activity on this for the production of single strain products or microbial consortia. The origin is always natural. They are usually isolated from the soil, natural soil, and we can have them as pure culture. They are isolated in the lab and they are produced in large quantity for the application. Regarding mycorrhiza, they similarly they can be isolated as spores. They can be extracted and cultivated in vitro or in plant roots, and then they are produced and commercialized as regarding the fungi. Usually they are in a form of granules, and they are together with roots usually. The bacterial strain can be produced as in a liquid form or in a powder form with some stabilizers. And the most important thing is that they can also be used as a consortia. So each strain may have its own properties. Altogether, they can have a synergic effect. And this consortia can be natural community, which are usually present in the soil, which usually are not so well characterized, while it's becoming more frequent to have the pure culture of single strain, which are put together according to their biocompatibility and produced in large quantity to be commercialized. So there are plenty of these products on the market. How can we use them? So they can be applied, always taking into account the 4R for fertilizer use, but they can be inoculated in different ways. So we have the seed coating procedure, which consists in putting them in strict contact with the seeds and cover the seed before, of course, before sowing. They can be applied in a liquid form, for example, on seed beds before transplant, which is common for horticultural crops, or they can be applied using the normal, let's say, procedure, using the machine the farmers have, and they can be applied before, during, or after sowing, always in a different formulation. Or it's also common to use them as a fertigation strategy, combined, for example, with precision irrigation. So we have a flexibility which allows the farmers to choose the best way of application according to their needs and according also to the crop. At the University of Parma, we had the possibility to make some trials with the different horticultural crops and cereals, sometimes in multiple years of application, combining the use of biostimulants with the reduction of fertilizers and with the precision irrigation strategies. Within the framework of different kind of project. I put just some example of European project which has been concluded so far, and on some regional project in Emilia-Romagna. And we could apply both commercial and research consortia, let's say, which maybe will become commercial in the future. So consortia, you can see some example here, just to give you an example of which are the properties. So we have a strain with the nitrogen fixation capacity, biocontrol capacity, plant growth promoting capacity. So we put together different strains which were compatible to grow together and which could give the plant different new properties. And we use also a lot of mycorrhiza, different kind of mycorrhiza, and applied in different way, as seed coating or straight to the soil. So we could test different combination. As an example of few results, I propose you field trial results. We made a field trial with the with the lettuce, and using mycorrhiza combined with a microbial consortium in a drip irrigation condition. We applied this product on the soil, and we verified that we had, among the many traits we tested both in leaf and roots, we observed an increase in fresh weight in leaf, and an increase of the stomatal conductance combined with a reduced leaf temperature, an increase in photosynthetic pigments and antioxidant metabolites, which were due to the increased root colonization by this fungus. We tested the recolonization because it's very important to test the efficacy and the real presence of the mycorrhiza. We measured the leaf temperature with a thermal camera, and we could see statistically also difference. This combination was tested in the greenhouse as well under controlled drought condition, and we observed also an improved stress resistance. In the case of tomato, we could test different combination of microbial consortium and mycorrhiza, also combined with biochar, but in this case it was used as a sort of a carrier and at a low quantity. We had 2 organic farm and 1 conventional in 3 years of field trials, and at the end of this experimentation, which was quite long, we observed an interesting increase of Brix degree on the fruits, a reduced apical rot, and a reduced amount of unripened fruit. So taking together all these results, we observed that the simultaneous addition of microbial consortium and mycorrhiza and char, we could obtain an increase in the commercial value of the fruit. So it's a sort of also economic evaluation, which in some cases is better in the conventional production or in the organic production according to the different combination. So this in terms of the utility, the practical utility. And we also tested some application of glomus as a mycorrhiza and trichoderma as well as a biocontrol agent on wheat during a field trial which was conducted in the Emilia-Romagna region in different locations. This was very interesting because we combined the reduction of fertilization by 60% respect to the recommended dose, and the significance of this result is double in the sense that we observe that reducing nitrogen fertilization doesn't reduce the production, but if we combine this reduction with the application of fungi and of trichoderma, we could increase by 11% the productivity. So there is a sort of double information in the sense that our soils, agricultural soils, are very rich of nutrients, and fertilization can be reduced effectively. But if we combine also the use of this product, of this biostimulant, we can still improve. And actually, these products work well at low dose of fertilization because they can be considered as a sort of complementation of the actual chemical fertilization, and this can help in reaching the EU Green Deal goals. So why I say biostimulants are a smart solution? Because they can improve adaptation toward the stress in general and can improve nutrition, giving higher yield, improved food quality, and decreased food loss as a consequence. And they also are a measure of mitigation because they can combine with a reduced fertilization, a reduced amount of pesticide, giving as a consequence a reduced impact on greenhouse gases emission and lowering contaminants in soil and water. The question is, which is the real impact? And the answer is complex because the impact can depend on the crop and sometimes even on the cultivar, on the different kind of products, on the original state of the soil. So this is very important to be able to assess the characteristic of the soil, to the crop management and to the environmental factor. Some meta-analysis are saying that the overall yield increase we can expect is between 5 and 10% in terms of yield, but we have to take also into account the downstream effect of reducing fertilizer and other kind of inputs that can give, let's say, improvement on soil quality at the end. It's very difficult to quantify this in terms of benefit, in the economic benefit. What is going on? There are many efforts about regulation. So there is, at the European level, the needs of harmonization of the regulatory landscape, the needs of standards for verifying the claims of this biostimulant. We need consistent guidelines for their production. And in the future, there will be also digital labeling for the fertilizing product in general and also for biostimulant. There are also some challenges that we have to face. So I will say the most important is the needs of research, a lot of research to understand the factors which influence the inoculum success. We need to know something about the shelf life of these products. We need to improve the delivery methods to be adaptable to all the practices used in the farmers. And this maybe will lead us to have higher acceptance by the farmers about the use of these products. So I will conclude with a few remarks. The biostimulant market is continuously increasing in Europe, so there is a lot of interest in this sector. There can be a smart solution, but more important, I think, is that we need more research in strict collaboration with farmers in real field condition, because we know a lot about the efficacy in the lab, in the greenhouse, in controlled condition, but we need more knowledge about their use in real condition. So I thank you for your attention. I'm open to questions.
Thank you, Professor Mariolina, and I invite you to submit your question if you have any regarding the previous speech or this one, or any question you have.